Hello friends, welcome back to my YouTube channel Nastin Padasalai. Currently we are revising for our GATE examination. So in this video we are going to study about Nobel Prize winners and their contribution to the field of microbiology. So first Nobel Prize winner is Sir Ronald Ross. He had done his contribution in studying the life cycle of Plasmodium in mosquitoes. Next is Robert Koch. He had discovered the causative agent responsible for infectious diseases like tuberculosis and anthrax. Next is Paul and Ellie McClintock. McClintock also Nobel Prize for discovering the types of phagocytes and the functions of phagocytes. Next is Paul. He got his Nobel Prize for discovering the types of blood leukocyte. Next is Carl Landsteiner. He got his Nobel Prize for discovering the ABO blood group system. And next is Sir Alexander Fleming. He got his Nobel Prize for discovering the miracle drug penicillin and its broad spectrum antibacterial action. Next is Selman A. Waxman. He got his Nobel Prize for discovering the drug called streptomycin, and he was the one who coined the term antibiotic. Next, these three persons, Beadle, Joshua, and Tatum, they have got a Nobel Prize for discovering the transmission of hereditary characters. Okay. Next is Hawley, Kurana, and Nirenberg. He got these three persons has done an immense contribution in discovering the genetic code and studying its function in protein synthesis. Next is Rosalind Yalow. She got his Nobel Prize for developing and defining the radioimmunoassay, which is used for detecting the hormone present in the tiny level. Next is Kerry Mullis. He got his Nobel Prize for his discovery of this polymerase chain reaction. Next is Stanley. He got his Nobel Prize for his prion discovery. Next, we are going to study about gram-positive bacteria and gram-negative bacteria. Okay, so first we'll be studying the gram reaction. So gram-positive bacteria, yeah, gram-positive bacteria, they will be retaining the crystal violet dye and they will be remain purple in color, whereas gram-negative bacteria can be easily decolorized and they will be accepting the counter stain and they will be staining red in color. Next is peptidoglycan layer. Gram-positive bacteria has a thick and multi-layer peptidoglycan layer, whereas gram-negative bacteria, they have a thin and single layer peptidoglycan layer. Next is teichoic acid. Teichoic acid is present in the cell wall of gram-positive bacteria, but they are absent in the cell wall of gram-negative bacteria. Next is periplasmic space, outer membrane, lipopolysaccharide are absent in gram-positive bacteria, but they are present in gram-negative bacteria. Next is with regarding to flagellar structure. With regarding to flagella of gram-positive bacteria, 2 rings are present in the basal bodies, whereas in gram-negative bacteria, 4 rings are present in the basal bodies. So this is the flagellar structures of gram-positive bacteria and gram-negative bacteria. In gram-positive bacteria, if you check, only 2 rings are present. First ring is MS ring, next ring is C ring. Whereas if you take gram-negative bacteria, totally 4 rings are present. One is L ring, next is P ring, next is MS ring, and next is C ring. Next is with regarding to the toxins produced. Gram-positive bacteria produce exotoxin and gram-negative bacteria produce endotoxin. So exotoxins are a protein that are produced inside the pathogenic bacteria as a usual process of their growth and metabolism, and these exotoxins will be released into the surrounding medium following the lysis. Next is endotoxins. Endotoxins are the lipid portion of the lipopolysaccharide, which are the part of outer membrane of cell wall of gram-negative bacteria, and these endotoxins are liberated only when the bacteria get died and the cell wall broke. So okay, don't confuse exotoxin and endotoxin. Exotoxin will be produced inside and endotoxins will be present in the cell wall of gram-negative bacteria. Next is resistance to physical disruption. Gram-positive bacteria are highly resistant to physical disruption because of their thick peptidoglycan layer, but gram-negative bacteria are low resistant, which means the physical disruption can easily kill the gram-negative bacteria due to the presence of thin peptidoglycan layer. Next is regarding to lipid and murine content. Since the gram-positive bacteria have a thick peptidoglycan layer, the murine content is very high and the lipid content is very low. Whereas if you take the gram-negative bacteria, the lipid content is very high because in gram-negative bacteria there is a presence of LPA, that is lipopolysaccharide is present. Okay, so yeah, lipid content is very high in the gram-negative bacteria, but the murine content is very low due to the peptidoglycan layer is very thin in case of gram-negative bacteria. So you should know these kind of details. And with regarding to cell wall disruption by this lysozymes, lysozymes are active in gram-positive bacteria. Okay, so lysozyme can exert their bacterial actions by hydrolyzing the glycosidic bond. Okay, whereas lysozymes are inactive in the cell wall of gram-negative bacteria. Next is regarding to resistant to drying. Okay, so gram-positive bacteria are highly resistant the process of drying. So if you take gram-negative bacteria is susceptible to the drying. The reason is that in gram-positive bacteria, the peptidoglycan layer is very thick, whereas in gram-negative bacteria, the peptidoglycan layer is very thick. Next, with regarding to antibiotic can easily destroy gram-positive bacteria, and antibiotic cannot able to destroy, or else gram-negative bacteria are more resistant to antibiotic. The reason is that in gram-negative bacteria, there is a presence of outer membrane, whereas in gram-positive bacteria, outer membrane is absent. So what this gram-negative bacteria will be doing means they will be having few pumps. Okay, through that pump, they will be pumping the antibiotic. So they can grow well even in the presence of antibiotic. Okay, so that's why we can't able to kill the gram-negative infectious bacteria with an antibiotic. Next, regarding to susceptibility to anionic detergent, since this gram-positive bacteria doesn't have the outer membrane, so they're highly susceptible. We can easily destroy the gram-positive bacteria in the presence of anionic detergents. Whereas gram-negative bacteria, they are more resistant due to the presence of outer membrane. Okay, so the next we will be studying the flagellar arrangement. Like first type is monotrichous, a single flagellum extending from the one end of cell. Okay, so you can able to see the diagram that I have given over here. Example for monotrichous flagella type is cholera vibrio. Okay, next is lophotrichous. So for lophotrichous, several flagella which is called as stuffed can extend from one end or both end of the cell. Example is spirilla. Next is peritrichous. Peritrichous means multiple flagella may randomly distributed over the entire cell. Example is typhoid bacillus. Next is amphitrichous. And in case of amphitrichous, a single flagella or multiple flagella can extend from both the end. Okay, so that's a difference between amphitrichous and lophotrichous. In lophotrichous, several flagella will be extending from one end or both end, whereas in amphitrichous, either single flagellum or multiple flagella will be extending in both cells of both end of the cell. Example is alkaligenous and alkaligenous fecalysis. Okay, so yeah, so we'll be continuing the notes in the upcoming videos. Thank you, friends. Thanks for watching this video.